Okay. So this is where we left off. So let's go ahead and dig back in. So prokaryotes, cell parts now. So when you take a look at prokaryotes, these are the simplest things we have, right? So we tend to study those. This is kind of a pattern we see all the time, right? We start with prokaryotes and we kind of use those lessons to learn more about eukaryotes. So what are some of the features of a prokaryotic cell? First of all, other than they're simple, which they are, they don't have any membrane-bound nucleus, right? They do have DNA, but it's basically housed in a region called the nucleoid region. So it's kind of like, just kind of like a little pile where you just sort of toss them in the cell, kind of like when you toss your dirty laundry in the southeast corner of your bedroom, right? And the clean laundry goes in the southwest corner of your bedroom on the floor, right? It doesn't necessarily mean that there's not a place to put it. It just means that there's not a pre-built structure, like especially if you don't have a closet to put them in. You also have a cell wall, ribosomes, right? Because you basically have to make proteins. And so if you're going to be alive, you have to make proteins at some point. And so the other main feature of this is you don't have any membrane-bound organelles like at all, right? So everything that you have is kind of like in one big giant room. And when you take a look at the prokaryotic cells, we basically split them into two domains. We learned about this in chapter 1. You have domain bacteria, which is kind of like E. coli and staph and salmonella, all those little critters we always worry about. That's basically bacteria. And then there's another domain within that, archaea. And archaea are still prokaryotic in the sense that they have a cell wall. They don't have any membrane-bound organelles. They don't have a nucleus, right? So all of those features of a prokaryotic are true for archaea. But with archaea, they're very distinctly different from bacteria, which is the reason why they get their own domain. So let's take a look at some of the structure, what sort of makes a bacteria, and we'll talk a little bit about the flagellar structure as well, and we'll kind of circle back around at the end of chapter 4, and we'll kind of pick that back up. So let's start from the inside, moving out. So first of all, you see you have cytoplasm inside the cell, but you see these little pink dots. Those are basically the ribosomes. Right. So if you're going to be living, you have to make proteins. If you're going to make proteins, you have to have ribosomes. So that's one of those necessary components for all living things. You'll notice also in the middle, you see this big ball of stuff here. That's your DNA. And the prokaryotic genome is actually a circular genome. It's a big circle. It's just kind of all flopped around and kind of folded on itself and just kind of piled up there in the middle. So that middle region where you put that DNA is called the nucleoid region. And there is a very kind of a distinctive place where you put it, even though it's not bound by a membrane the way a nucleus is. As you continue to move outward, the first layer you're going to run into is the plasma membrane, a lipid bilayer, just like we learned about in the last chapter. And then outside of that, you're going to have a cell wall. We're going to take a look at the construction of the cell wall here in just a second. And then beyond that is going to be the capsule, which is kind of like a gelatinous sort of a layer, has different types of consistencies to it, but it's a fairly thick layer. And basically the cell wall and the capsule are there for the purposes of protection. So this is basically kind of a protective hide, if you will, a protective husk that protects you from the outside environment. Remember, the life of a bacterium is not for the timid, because everything wants to kill you. So as a result of that, living amongst enemies like your entire existence, you tend to sort of have multiple layers of armor. And that's the reason why you have a cell wall for protection and a capsule for protection. Now, to this capsule are going to be these hair-like extensions on the bacterium itself. And those are the pili, pilus for singular, pili for plural. And these are like little hair-like extensions that will basically allow the bacterium to interact with other bacteria It'll also give you sort of like something to sort of tether or anchor yourself onto a hard substrate. As a matter of fact, bacteria have the ability to, they don't really go through sexual reproduction, but they can actually transfer information from one bacterium to another through a process called conjugation, which basically it's you take a pilus and you kind of call it the conjugation pilus, and it's basically kind of creates like a little connector between the two bacteria, and they're able to swap stuff back and forth. Now, what's not on this that I want to make sure I highlight to you guys is another little thing that's inside bacteria. It's usually like a little circular piece of DNA. That's called a plasmid. It's very small, and it's a tiny circle. But these plasmid like DNA rings, little rings of DNA, are actually very easy to swap around. So a bacterium can exchange or swap these plasmid DNAs between each other, but not their genome. That's typically a no-no. And typically what you find on these plasmids are things like antibiotic resistance genes. You'll also find mating types. So they don't have gender. So they don't have like male, female bacteria. They have what's called mating types at F and F prime. But like an F prime mating type would conjugate with an F mating type. So kind of is sort of the preliminary gender kind of an idea. But those mating types would be on some of these plasmids. And there's other genes on there as well. And so these plasmids are highly, they're highly variable. So you can add and subtract things onto it very easily. And you can swap them back and forth between your bacterial partners. And it sort of represents a really kind of a cool system, so that you have basically you're stuck with your genome, right, for good or for bad. But then you kind of have this little outlet where you can kind of swap these little cassettes of DNA back and forth. And so that's basically your bacterial cell. If you recall, when we looked at the scanning electron micrograph images, remember the little furry guy we saw? That's a bacterium with its little pili. It's all that fur, those little pili, fibers that are sticking off of it. So let's take a look at some of the structure. We're not going to get too deep into this because micro, which actually happens in this room, would go into the deep details of this. So if you're going to be taking micro, you're going to get plenty of detail in terms of bacterial biology. But when you take a look at bacterial cell walls, the one thing you want to understand is they're not constructed the same way as the cell walls that we see in eukaryotes, right? So for instance, in bacteria, the cell walls are going to be made of what's called a peptidoglycan. So this is basically a protein peptide, right? That's where the term comes from. This is a protein carbohydrate fusion molecule. So it's kind of like part protein, part carbohydrate. And it forms the major structural component of the bacterial cell wall. Now it's different than in plants and fungi, right? Because what is a plant cell wall made out of? We learned about that one in the last chapter. It's one of the structural carbohydrates. What? Back in here. It's a polysaccharide. Which one? You eat it all the time. You can. What? Nope. You eat that as well, but that's not the structural. Those aren't the structural polysaccharides. Remember, we had 2 nutritional polysaccharides and we had 2 structural polysaccharides. Which one composed the plant cell wall? We actually saw a scanning electron micrograph of one of these fibers from the plant cell wall. Ooh, it's a good thing the exam's not till next week. Something we can't digest? What? It's fibrous, but it's not fibrinogen. You're thinking your AMP is leading you astray there, because you're going AMP with the fibrinogen one. So plants basically are composed of a structural carbohydrate and fungi as well. So what were the two? The first one is cellulose, right? So basically plants are made out of cellulose, and fungi is made out of okay. So basically, because you got cell walls, you can fill up with water and not blow up, right? So that's basically kind of protects you against water, basically taking on water. This is also the target of our antibiotics, right? So the cell wall, because it's so unusual, it's not like any eukaryote, we actually use antibiotics to go after these cell walls, and different components of the cell walls will be susceptible to different types of antibiotics. Right? And so oftentimes we can actually categorize different bacterial cells according to their cell wall structure. And that's typically how antibiotics work. They basically go after some unique things in the bacterial cell wall and disrupt some important biology in the bacterial cell wall, and that kind of blows the bacteria up and kind of kills them off.
It's like a type of HIV, coccus or something like that. It has like a cell wall, and you have to take an antibiotic because your immune cells can't get rid of this.
Yeah, because usually your immune cells, they'll attack the bacteria because the bacterial proteins are not common to humans, so they see them as foreign. And then they'll attack the bacteria. But if you don't have the immune cells to be able to recognize the bacteria, then you need a little bit of extra help. So normally, like bacteria that we wouldn't have a need to take antibiotics for because our immune systems are good at recognizing them and killing them. If you're immune deprived or immune deficient, like in HIV, for instance, then you have to have extra medical help. So your antibiotics in that case would be covering for the deficiency of your immune system. And that's a common thing that we see, because there's not just HIV, but there's other immune depressed and immune insufficient situations as well. And then of course you wrap all that around with your jelly-like capsule. Okay, so let's take a look at archaea, right? So bacteria, we talked a little bit about what's the deal about archaea, right? So how come they're prokaryotes but then not bacteria? Well, they're prokaryotes because they have a cell wall, right? They don't have any organelles, they don't have a nucleus. So all those things that are true for prokaryotes are also true for archaea. That's why it's a prokaryote. The reason why it's not a bacterium is because there's a lot of significant things that are very different at a very fundamental level from bacteria. For instance, the cell wall for archaea is not made out of peptidoglycan. It's a different type of polysaccharide protein construction, but it's not a peptidoglycan specifically. So its cell wall is actually a different structure than you would see in bacteria. Also, the lipids are distinctly different between archaea and bacteria, the types of lipids that they use in their membranes. But really the kicker is in the processes that they have. So for instance, when we started studying DNA replication and protein synthesis, both processes that we'll be studying toward the end of the semester, what we saw in archaea was something that is distinctly prokaryotic, right? No nucleus, no organelles, a cell wall that's not a eukaryotic cell wall. But when you looked at its processes, when you looked at like replication and synthesis, the actual mechanism that it used looked very similar to the way eukaryotes did business. So prokaryotes do replication in a very different way than eukaryotes, but archaea do it in a very similar way to eukaryotes. So in some very fundamental ways, archaea look kind of like eukaryotes. In some cases, they look kind of like bacteria. So if you want to think about it, and it helps you, you can almost imagine the three domains as being kind of like a Venn diagram that overlaps, right? So bacteria would kind of be over here on its own in the prokaryotic world. Eukaryotes would be over here, and in the overlap, you would have archaea, still prokaryotic, but kind of an overlap. Some things bacterial-like, some things eukaryote-like. Okay, so let's take a look at one important structure of the bacteria, the flagella, right? Because the flagella is necessary for locomotion. Got to have this one. So first of all, when you take a look at this, bacteria have flagella. They can have one or two or three. They can be in all places. They can be multipolar. So you could have a bacterium that could have one flagellum. You could have a whole bunch of them out there. Some will have flagella on either end, and they'll have multiple on either end. So you have lots of different variations of where you can put a flagellum for a bacterium, because for a bacterium, you don't have like a head and a tail. You don't have a front and a back. So this predates symmetry. So you don't have like body axis symmetry issues. Chris?
I was going to ask, is it more in the sense of like similarity to whiskers on a cat or a dog versus
No, really, it's good, completely different than whiskers. Whiskers are all sensory. Whiskers are basically modified hair. That's basically what whiskers are, that are modified to be in contact with sensory organs. And that's the way they feel. So they actually feel with their whiskers. And so they're like sensory structures, which is the reason why if you shave off an animal's whiskers, that's kind of a little on the mean-spirited side. That's kind of like shaving off part of their nervous system. So this is all locomotion. So what we're going to do is we're going to take a look at the structure of how bacteria construct this and how they move. But the basic physics of it is pretty much the same thing as a boat propeller, right? So basically you have a boat propeller and it's a rotary motor. Basically you sort of spin this thing in a circle, and what it does, it creates basically a push. It pushes against the water and moves the boat forward. And the same thing is also true for bacteria. Now, one of the things that I want to make clear is the source of energy for this particular system. It comes from a proton gradient. Now, what does that mean? So one of the things you want to make sure that you understand clearly is that a proton is exactly the same as a hydrogen ion. In acid-base discussions, we talk about hydrogen ion because we're talking about donating the hydrogen ion, the increase of hydrogen ion, because that's what you measured, right? Hydrogen ion. But in reality, there's no difference between a proton and a hydrogen ion. As a matter of fact, when you're talking about cell physiology, like we are now, you typically will oftentimes refer to proton gradients, not hydrogen ion gradients. Okay. Why? Well, because let's take a look at what's in a hydrogen atom, right? A hydrogen atom has a mass of 1. It has an atomic number of 1. So how many protons does hydrogen have? 1. How many electrons does it have? How many neutrons does it have? If its mass is 1, it has how many neutrons? Because it already has 1 proton. So how many neutrons does it have? None, right? So all a hydrogen atom is 1 proton, 1 electron. So what happens when you take that electron away to create your hydrogen ion? You literally have a proton left. So a proton is exactly the same as a hydrogen ion. So what is a proton gradient? Well, basically a proton gradient is an energetic strategy that we actually use a lot in energetics in the cell.
It's basically the same set of physics as a hydroelectric dam. All you're doing is creating an energy gradient, right? Where you have a high energy versus a low energy side. And you utilize that in your favor. So basically what you have, like the hydroelectric dam, if you take a look at Hoover Dam, for instance, you have a high water column behind the dam, you have a low water column in front of the dam. Well, this is going to be high in energy, yes? How do you know that? Because if you break the dam, you stand in front of it, you're going to see just how much energy is coming at you, right? So there's a lot of energy there. And of course, energy wants to flow from a high to a low source, right? And so what we do in the dam is we say, okay, fine, if you want to flow from high to low, we will let you, but you have to go through the dam and you have to turn our turbines, which are basically copper wires and magnets spinning around each other, which we all know is basically sufficient for converting and creating electricity, which will light up Las Vegas, right? And so we basically use the energy built up behind the dam to essentially create electricity. And so it's an energy conversion. You do the exact same thing on a cell physiology. You use a proton gradient, which is built up on one side, and as it flows down its energy gradient, it releases energy, and we use that to do some work. So what kind of work are we doing? Well, let's take a look at it. So let's take a look at the bacterial wall construction. The first thing you're going to run into is the plasma membrane, and inside the plasma membrane is going to be embedded two ring-like proteins that are rotary. They're rotational, so they can rotate. And so there's going to be two of these little circular protein drums that are going to be embedded in the membrane. Then you're going to have like a little bit of a shaft structure. And then you're going to have another set of rings embedded in the peptidoglycan wall, which is this region here. And then the outer wall, there's a little bit more than just peptidoglycan. There's another outer external layer of the wall complex, if you will. And you have an outer ring embedded in that area there. So this whole thing together is kind of like the wall complex. And then the shaft continues to go through those two rings like a pair of dumbbells, right? And then what happens is it turns, it bends, it curves into this sort of filamentous sort of long structure, which is the flagellum. Now what happens? So what happens is you basically take the proton gradient, which is high on one side, low on the other. And just like with osmosis diffusion, we know that everything flows from high to low. So what's going to happen is your proton is going to basically flow from high to low. As it does so, it's going to release energy, and that's going to basically give you enough energy to rotate these little drums. And as they rotate, they're going to spin that little bent fiber that's on the outside of the flagellum. And so basically you're going to get this sort of rotating flagellum that will just basically kind of rotate just like a propeller in the water, and it'll basically it'll push against the fluid in this direction, which propels the bacteria in the opposite direction, right? So just like a boat propeller, the boat propeller is pushing on the water in that direction, your boat basically, right, equal and opposite reactions, your boat moves the opposite direction. And so that's the bacterial flagellum. So that allows bacteria to be able to move and to be able to swim around, which is really important, right? Because they have to be able to find food, and they have to be able to find resources, and they have to be able to move around because they have to go looking for that. Okay. Now, what about eukaryotic cells? Well, what's the deal with them? What sort of sets eukaryotic cells apart, right? Because it seems like prokaryotes kind of have a good gig here. Well, first of all, one of these things that sets them apart is going to be the nucleus. So the nucleus is obviously one of the big ones, and of course, all the other membrane-bound organelles goes along with it. This basically gives the eukaryotic cells much more complexity than prokaryotic cells, and it allows them to be able to compartmentalize their functions into membrane-bound organelles. So what does this do? This gives you division of labor. And organization of output. Many of us have been in workspaces where there's not a lot of organization, like actual physical organization. And we've oftentimes made note of like, oh, well, this is not really a productive environment because, you know, I don't have what I need, right? And then some of us have been in workspaces where there's a lot of organization. We realize just how functional that is. Here's a good example. If we walk into an empty classroom, you're going to have a certain amount of functionality there, yes? but are you at your most functional if it's just an empty classroom, zero furniture? No. What do I need to do in order to make you extra functional? Put desks in there, tables, places for you to sit down, workspace. So now all of a sudden you've got an organizational tool to help increase your productivity. All I did was compartmentalize you. If you get your own little workspace, your own little desk, then you've got something to basically put your notes on so that you can write. You've got a place maybe in that desk to store stuff, like like a high school desk. Basically, you kind of stick stuff underneath. So you have different areas, different compartments with different functions. All of them together gives you maximum functionality and maximum productivity. Notice all I did was organize the space. That's all I did, and create organizational structure, the desks, to help you organize your output. Now all of a sudden you're much more productive. Same thing's going on with the eukaryote. So when you take a look at your division of labor, you basically compartmentalize all of the activities of the cell and you put them in specific organelles. Typically is to answer a biological question of life. Not all of them, but one of them, right? Because the idea is if you put all of them together, then you've got all your questions covered. And as a living cell, you are the basic unit of life, right? So you can answer all of your questions. So one of the big things we're going to be taking a look at is an innovation of eukaryotes called the endomembrane system. Okay, so we're going to take a close look at that one. The other one that's also a big innovation for eukaryotes is the cytoskeleton. It's basically the skeleton of the cell, which sounds kind of boring, but prokaryotes don't have it. Eukaryotes do. And actually, it gives you a lot of functionality. So we're also going to talk a lot about the cytoskeleton and the role of the cytoskeleton. So what are some of the features of your eukaryotic cell? So what are some of the things that they have? Well, they have a plasma membrane, but so does everybody else, right? That's one of the things that's on our list of all cells needing to have a plasma membrane. Cytoplasm, that's also another one that all cells have to have, right? Organelles, that's something that's unique to eukaryotes. Cytoskeleton, we just talked about that one, that's unique to eukaryotes. Nucleus, that's unique to eukaryotes. So you can see you have some things that are the same as prokaryotes, right? Do prokaryotes have a plasma membrane? Yes, they do, because all cells have to have one, right? Do prokaryotes have cytoplasm? Well, yes, they do, because all cells have to have cytoplasm. But where the innovations come in are the organelles, that compartmentalization and the division of labor and the organizing of your activities, the cytoskeleton and the nucleus. Those are the biggies. Okay. So that brings us to this one, where typically your heart stops beating for a second or two and you're not breathing anymore because there's only one question in your mind. Do I have to know all that? And unfortunately, the answer is yes. Now, what this is, and I want to stop a little bit and sort of explain what this is and give you a little bit of tips here, just to kind of give you some survival strategies. You'd be wise to pay attention. This is one of those images in a chapter that sort of pulls together large swaths of the information in the chapter. One of the biggest questions I get from a chapter like this, if I haven't already gotten it, which I have gotten it already from some of you guys, is how in the world do I study for this chapter? Well, first of all, how you study for anything is to index the knowledge. Right now, if you're just going slide by slide, organelle by organelle, we're talking about a structure and its function, which is basically a pretty common set of questions in this type of a chapter. What is it? What does it do? Right? Then all your brain is basically picking through a weed patch of random chaos. Right? So when you sit down to an exam, it's not going to go well, right? Because you're asking your brain at every question to pick through this sort of random, sort of trashed basement of factoids that have absolutely no consistency or cohesion between the ideas. Don't do that to your brain, right? This is one of the reasons why I say one of the best things you can do when you have a high volume of information is to figure out a good strategy to index the knowledge. One way to index the knowledge is like this. First of all, our dominant sense is our sight, which means we are very visual. Even those of us who claim not to be visual, we're very visual. 9 times out of 10, on an exam, the first things that you recall are images, pictures, right? That's just the way we're wired. So what this kind of a slide does is it basically uses what your brain is already naturally inclined to recall to your advantage, using an image as an organizational tool to basically put information onto it. So one of the things I always tell students to do, use something like this to study the chapter. Because when you're at an exam, you're more likely going to recall this image. And if you've studied all the information on this image, then your brain will basically pick out the image. And then a lot of times the information that was on this slide is associated with the image will come along with it. And so you can use the image as sort of like a magnet to pull the information out of your brain. It's there, you just have to find it. This is a way to sort of use it as a magnet to pull that out. Oftentimes, the more information you can put on a slide like this, the better chance you have of recalling information. Why? Because first of all, it does a couple of things. It's visual, so it's easier to recall. Second thing is it's orderly. So you can see you've got a structure and you've got a little bit of a functional definition. Now, what I would do is as you're studying through this, we're going to unpack each of the organelles in step as we go along here in just a second. But as we go through this, I would take this and I would add lines of detail. So study the image, get comfortable with it, and then add a line of detail. And then study it again, and then add another line of detail. The more times you do that, the more lines of detail you add, the more information you're sticking to this image. So on the exam, when your brain pulls it out, you're going to be pulling out more information per question. And the more information you can pull out, the more questions you're going to be able to answer correctly. It basically also represents a visual table of contents for most of the rest of the chapter. Most of the rest of the chapter is basically outlined here. We're going to go organelle by organelle, break it all down, and sort of determine how it's structured and what it does. Okay. And there's a couple of other topics at the end that we'll cover outside of this, but that's what this is. So this covers an enormous chunk of the chapter itself. Okay. So use this to your advantage. There's another one also, right? The plant cell. How do we know that the plant, what sticks out? Not so much the shape, not necessarily the green. There's specific things in here that betray it as a plant. Wall, right? The cell wall is what sticks out. Animals don't have a cell wall, right? What's another one? Big pool in the middle.
The central.
The vacuole.
Yeah.
Yep. Right. Animals don't have that one. There's one other one. Chloroplasts. Right. That's where photosynthesis is happening. So they happen to be green, but it's not the green that makes it a plant. It's the chloroplasts that make it green. So that's the actual organelle. Everything else is the same. Notice it's got mitochondria, it's got all the same organelles, right? So there's only those three that really kind of stick out. And so we're basically going to be going through the rest of the chapter, sort of breaking all these organelles down. Now, what I'm going to do is I'm going to do another organizational tool, another indexing strategy. That is another one. Yeah, that'll be on the list. That's the reverse, right? Animals have that one, plants don't. So that's definitely true. Yep. So, so this is like a visual index, right? It kind of puts everything there. And another reason why this is a really good one is I know a lot of you guys are flashcard people, right? And I'm not necessarily saying don't do flashcards, but there's a strong weakness to flashcards because basically all flashcards are good when you are trying to study definitions, right? Not when you're trying to study process, because process, things that have context to them, flashcards don't capture that context. Okay, so for instance, you can run down all the organelles and be like, this does this, this does this, and that'd be good. Right? I'm not saying don't do that, but don't do that only, because that doesn't give you the context. The reason why this is a good image is because it shows you what everything is, what it is, and what it does, but it also puts it into its proper context, so you understand where it lives and what part of the cell it works in, because that's the piece that we want to get to, is that functionality component. Okay. And biology is a very context-driven discipline. And flashcards tend to lose context. That's the reason why flashcards tend to be a weaker way of studying for things like biology. But still though, right, there's a lot here, right? How do we choose which one to start first, right? Which one do we start with first? Well, I'm going to give you another organizational tool that is something you can use anywhere, anytime, in any way. And it does take time to sort of flesh it out. I'm going to go fast. So you will want to make sure that you revisit this on the recording, because I want to get it out there, because it's basically an outline of the organelles by function, grouped by function. There's 4 major global functions that you can throw most organelles into. So let's go. The first one is the informational organelles. And this starts off with the nucleus, which basically houses your DNA. Within the nucleus, you're going to see the nucleolus, which is essentially an aggregation of ribosomal RNA. We'll take a look at that in the chapter a little further down the stream in the chapter. That's a structural component of ribosomes, and ribosomes are the next one on the list, right? So these are basically protein complexes that make protein, right? That's another one of those evolutionary chicken egg things, those little circular reasoning things, right? A protein that makes a protein. So who makes the protein that makes the protein, right? That's one of those things. So those are your informational organelles. We'll start with those guys first, and then we're going to go into the big innovator of eukaryotes, the endomembrane system. And this has a series of organelles. And so we're going to basically study the endomembrane system through a mechanism, a process called the secretory pathway. And basically what the secretory pathway is the production of a protein and the release of that protein to the outside of the cell, typically for the purposes of communication and coordination with your neighboring cells when you're working together as a team to do some specific function. The first stop is going to be the endoplasmic reticulum. Which has 2 different types. You have rough ER, which is involved in protein synthesis. And you have the smooth ER, which has 3 main functions. It has a detoxification function, a calcium storage function, and lipid synthesis. Then from there, we're going to basically take a look at transport vesicles. These guys are basically membrane bubbles full of crude protein. So they're going to the next stop. The next stop would then be the Golgi apparatus. Which basically does 3 main things. It will modify that protein, it'll sort it, and it'll ship that protein. And then that'll package that into a secretory vesicle. Which is a membrane bubble full of finished protein. These here represent the secretory pathway. We'll talk a little bit about that. Now, an extension of this, which I tend to put under the endomembrane system, is going to be a couple of other organelles that we'll take a look at.
Basically the lysosome, which is the garbage disposal of the cell, and the peroxisome. Which detoxifies oxidative byproducts. So that's number 2. Big one, important one. Remember, the endomembrane system is one of the eukaryotic innovations, right? This is a big deal. So number 3 are the energy-related organelles. And you have really 2 of them, right? You have mitochondria, and their job is to make ATP. And then you also have chloroplast. And the job of chloroplast is photosynthesis. And we have entire chapters for that one. So I'm not going to dig into that one too much, even in this chapter. I'm not going to dig into that one too much, because the actual nitty gritty of what's going on in mitochondria and chloroplast is unfurled in chapter 7 and chapter 8. Okay. The last one is the other big innovation, and that's the cytoskeleton, right? So this is basically the support structure of the cell. And there's basically 3 main fibers. There's microfilaments, intermediate filaments, and then there's microtubules. 3 different fibers in the cytoskeleton, and they have 3 distinct different jobs. We're going to see examples of all 3 of them, actually, and unpack some of the biology that they are responsible for. So that is the 4th main. So there's 4 main functional groupings for all these organelles. And then after that, so that's going to be a lot of the rest of the chapter that we're looking at, which is the reason why that picture of the cell is like a little visual table of contents for everything we're going to be covering. And then after that, there's going to be a couple of other topics that we'll talk about as well. And that'll be largely the end of chapter 4. Okay, first off, our informational molecules, the nucleus, right? So the nucleus is going to be the repository. It's going to be the place where we hold our DNA. Most of our cells will have a single nucleus. There will be some with multiple nuclei, but most will have single nuclei. So that's going to be just where we house our DNA. Now, inside the nucleus is going to be a non-membrane bound region called the nucleolus. This is an aggregation of ribosomal RNA, right, which is basically a structural component of the ribosome. And then it's all bounded by 2, not just 1, but 2 lipid bilayers, a nuclear envelope with an inner and an outer layer. And then within that, you have nuclear pores. So you have a nucleus with 2 membranes, and then you have pores in them to allow passage of molecules into and out of the nucleus. But remember, this is restricted passage. So not just anybody can get into the nucleus. If you're in the nucleus, it's because you got something to do there. You don't just get to blow on in there, right? Think of the nucleus as sort of like the Oval Office, right? We say it's like, oh, well, the White House is the people's house, right? Well, yeah, you try go cruising into the Oval Office and see how far you get, right? So yeah, you're going to get thrown on the ground. Your entrance to the Oval Office is by invitation only. And that's true for not just Joe Average like us, but that's true for pretty important people, right? Like the leader of the Senate, for instance, like Schumer, who's actually a member of the president's own party. Can't actually just go breeze right on into the Oval Office. Can't do it. Speaker of the House can't just go breeze right onto the Oval Office. Only the people that the president wants in the Oval Office gets to go in the Oval Office. Why is it so restricted? The Illuminati. Okay, minus conspiracy theories. Why is it so constricted? Yeah, what are we securitizing? What are we securitizing? What are we protecting?
Okay, the big red button, right? But what else is in there that we're protecting?
Are you going to have it, like, indicate that
Yeah, right. I mean, and it doesn't matter who the president is, whether you like him or not. You can't get into the Oval Office, right? Because ultimately you're trying to protect the president, right? So, right. So that's true for any president. You can't just breeze right on in there. And so there's obviously a lot of stuff in there as well, right? There's a lot of information that's classified and things of that nature. But there's a reason. There's information in there that's not to be heard by others, right? I mean, this is typically where most presidents get their daily intelligence debriefing, right? When they're learning about all the secrets of the state that the CIA is dredging up across the world that Joe Average shouldn't know about, right? I mean, these are really top secret meetings in here. I don't know if you've ever seen the White House or the Oval Office wall, the door. It's like ridiculous. It's like, what, about 20 feet thick? It's like ridiculous. It's not a door, it's like a wall. Yeah, it's like crazy. But that's because there's a lot of stuff in there that you're protecting. You think about anything that a president is working on at any given point. They could have all sorts of very sensitive classified information strewn all over their desks that they're working on, trying to figure out what to do with this country, or, you know, what's our policy on this one, because the president's the one who has to make all those decisions. And so there's a lot of sensitive information. Just like with your nucleus, there's a lot of sensitive information in your nucleus. After all, that's all the instructions that you need to make you from scratch. That's pretty sensitive stuff. You screw that one up and you are toast, right? Question. Yeah, I mean, it's the same, right? I mean, it's not easy to get into nucleus, but you can. But the people who are getting in the nucleus have business to do there. Yeah, you don't get to go sightseeing in the Oval Office usually, right? That's not something you get to do. So it's restricted. And then, of course, with eukaryotes, instead of being circular, our genome is not circular like bacteria are. It's linear. And actually, our DNA doesn't exist as just naked DNA. A lot of times students think it's like, oh, well, this is kind of what the DNA looks like inside our nucleus. That's not true, right? And the reason is very simple, because the DNA molecule itself is very delicate in structure. So if you were to grab onto that molecule and pull on it, it would be like grabbing onto a cobweb and trying to move it. It would just disintegrate, okay? So what we do in order to protect it was we put in a structure called chromatin, which is a combination of the chromosome and proteins that are associated with it. So what we have are little drum-like complexes of protein called histones. So there's 8 histone proteins that form this little drum. And we have several of these little drum-like complexes. And around it, we wind our DNA. So this structure is what chromatin is. Matter of fact, each one of these individual units we refer to as a nucleosome. But the actual structure of DNA in the nucleus is kind of DNA wound around these little protein drums. Okay. So now this is what DNA actually looks like. Here's the structure of it. So first of all, notice you've got these nuclear pores in your otherwise double membrane system. So if you take a look down, here's a blow up of your nuclear envelope. Notice you have an inner envelope, you've got a space, and you've got an outer region. And then basically it's punctuated by these nuclear pores. So you can see that you got these proteins that are basically embedded in the nuclear envelope that kind of creates these little circular pores. And that's so you can get traffic coming in and out. But it's not just an opening. You have these structures. You have this nuclear basket, which is this kind of protein attached to these little kind of fibrous. And what this is called the nuclear basket. And then you have other cytoplasmic extensions that sort of extend inward and sort of attach to other things that are inside the nucleus. So it's kind of like an anchorage sort of a system. But this also allows you to be able to regulate transport into and out of nucleus, right? So this is kind of like where you would check your credentials. You want to get into the nucleus? Let me see your security clearance, right? and you got some, hired goon there who's about ready to rip you apart if you don't have the right security clearance, right? Because they automatically assume you're there to kill the president. So they just take you down. So on the other side, though, you also have some organization, right? You have the filaments of protein that sort of extend out in the cytoplasm that anchor the nucleus to other external features in the cytoplasm itself. Then as you kind of move inward, you see this sort of marbly looking haze. It looks like a bowling ball, right? So this kind of marbly looking haze in there, that's what's called the nucleoplasm. That's fluid plus stuff, right? And in that you can see there's chromatin. That's where all your DNA, your chromatin, your chromosomes are, and where they're kind of doing their job. And then this whole structure right here, which is rather unfortunate, but because of 3D computer graphics, this is the way they do it. It kind of looks like a little bowling ball. These edges are a little more defined than they actually are supposed to be. So this is the nucleolus. Okay, so the nucleolus, remember, is just an aggregation. It's a pile of ribosomal RNA. As a result, the edges are not so well defined. All right, so this is a little bit misleading oftentimes for students. One of the common mistakes that I see students make is they basically take a look at this and they say, oh, well, that's the nucleus. No, it's not. That's the nucleolus. The nucleus is inside the nucleus. And it's typically ill-defined. It's not a membrane-bound system. It's just a pile. It's just a pile of ribosomal RNA because you're busy making proteins. You're busy constructing ribosomes, the factories that make proteins. Okay, question? It's these, this is not, well, it's not really purple. It's actually black and white, but they don't have a color. So these are all colorless. We just kind of make them pretty to be appealing because we know that the more pretty it is, the more likely you're going to remember it. So a lot of the colors are just fictitious. Most electron micrographs are black and white. And so these electron micrographs, then you'll see, this is like a scanning electron micrograph here. You can see the surface of a nucleus. So you don't just have a pore here and there. You've got like tons of pores, right? So there's a lot of regulated traffic going into and out of the nucleus. I mean, it makes a lot of sense, right? The information that's in there is a working document, and you've got a lot of proteins that you're making in the cell. So you would imagine that your DNA is going to be in hot demand because you're constantly having to make proteins. One second. Sure. Right. It all depends on how much you're making, right? So like, for instance, if you're not making that much protein and there's not a whole lot of demand for the information that's on the DNA, then you don't really need a whole lot of nuclear pores because your activity doesn't support that. It can change from cell to cell. Yeah, you can add them and subtract them, but usually you don't. I mean, usually there's just so many proteins that you have to make to do all the work of the cell that more than likely you're going to see something that looks like this because there's just a ton of protein to make. So the other one is going to be a transmission electron micrograph. So here you can see the actual double membrane system of the nuclear envelope. So here's your cytoplasm. This is your nucleus side. And you can see you have your outer and then you have your inner here. So you can see those two pieces of the envelope, and then they kind of come together in a pore, and then they kind of exit again into this double membrane system. So that's a very good view of this nuclear envelope system. Now this guy down here is the nuclear lamina, and this is also a scanning electron micrograph. Now the nuclear lamina is going to be this little orange line that's right underneath the inner membrane of the envelope. Now, what is the nuclear lamina? So the nuclear lamina is basically an intermediate filament made of the protein laminin. So basically you have lots of laminin that kind of forms these little filamentous chains. And then those chains will then crosshatch into this kind of gauze-like structure just underneath the inner membrane. So it's very similar in nature and structure to like that kind of gauze-like material when you do casting and you kind of dip the gauze-like material into the casting compound, and then it sets up hard when you wrap that around the individual's arm. Yes, Betsy.
What is that?
Which one?
Under the-
Underneath? So it's an intermediate filament made of the protein laminin. So this is an example of the cytoskeleton, right? So what does the nuclear lamina do then? So the nuclear lamina basically gives the nucleus its shape. So without that nuclear lamina just underneath the inner membrane, the nucleus would just basically deflate and it would collapse down on itself like an evacuated balloon.
Arm it, or is it more visual?
Well, yeah, that'd be a functionless nucleus, right? Because everything would just collapse down on you, and then you wouldn't be able to do your business, right? Think of these intermediate filaments. In a lot of ways, you can think of them as sort of like the framing rods of a tent, right? The framing rods of a tent are designed to basically create the basic framework and the shape of the tent, and that's what keeps the tent fabric up, right? So if you lose those framing rods, then basically you just have a bunch of fabric that just falls down on you and you're just kind of wallowing around in sheets, basically, right? And so the intermediate filaments, in this case, the nuclear lamina, does the same thing. It's basically keeping that circular structure of the nuclear envelope open so that you can do all your business on the inside of the nucleus itself. Perfect example of the role of intermediate filaments as a cytoskeletal support structure, as a scaffold that creates shape and structure for the cell. Okay, let's take a look at ribosomes, right? So these guys are the factories, basically, that make proteins. And so you typically have ribosomal RNA as part of that complex. So that's the structural component of it. That's the reason why if you have a nucleolus, you know that you're busy about making proteins. That's kind of something you're doing. But the entire process of protein synthesis, which we'll talk more about in the details a little bit later on, actually encompasses all three major types of RNA, right? So ribosomal RNA is a structural component of the ribosome, which is essentially the factory for protein synthesis. But mRNA, this one is going to be a copy of the code, right? When we talked about mRNA being a copy of the DNA, well, that's mRNA, right? So it's got the information necessary to pick the correct amino acids in sequence. And then transfer RNA is actually a piece of RNA in protein synthesis that allows you to be able to physically grab that correct amino acid based on whatever the code is on mRNA. So you see all three of them. We'll actually take a much closer look at the role of all three once we get into the last chapter of the semester. So I don't want to bog down too much on that one here. The important thing about ribosomes is you can basically organize them pretty easily, right? So for instance, you can either have ribosomes free out in the cytoplasm, or you can have them actually bound to internal membranes, which is very helpful, right? Because think about it. If you're basically going to be making a protein whose job is located in the nucleus, does it make a lot of sense to send this particular ribosome to make this protein way out in the back 40 of the cell? If it has to come back to the nucleus to be functional, why would you send it out to the pasture to be made? Because then you're just going to figure out some sort of a distribution mechanism to bring it back in, right? So the better idea would be like, why don't you just tether a ribosome to the nucleus and have that ribosome specialize in just making those nuclear proteins? That way, once it's made, it's already there. It's on the job site. It just has to get to work. Okay. So you can actually organize these ribosomes based on, and you can sort of categorize them based on what proteins they make and where they make them. So you have a lot of organizational capacity there. Bacteria don't do that because they don't have any internal membranes, number one, and everything is just sort of all out there in the cytoplasm, right? So they just kind of make it wherever. So that's a big thing. So the ribosome has two main subunits. It's got a large subunit that then clasps onto a small subunit during assembly, and that's also part of the protein synthesis component. We'll talk more about that in the last chapter of the semester. But then it's ready to roll. So that represents the end of the first major group, the informational organelles. Now for one of the big innovative groups, the endomembrane system. So why do we call this endomembrane system? So endo means inside,
right? So this basically means inside the membrane. So what we mean here is basically a protein can be made and released without crossing a membrane. That's why it's called the endomembrane system. And so ultimately then what's happening is you've got a series of membranes, a series of organelles, stopping stations, where you have transport between each of these major sort of organelle compartments. Think of the endomembrane system as like an assembly line, right? In an assembly line, if you've ever gone on a factory tour, you notice that in the factory, they've got different stations, right? So like, this is our product development area. This is our packaging area, right? This is our, they got different things going on at different areas in the major overall assembly line. And that's what this is like, right? So you have in the process of producing a protein and getting it to be functional and then releasing it, there are different stopping points in this assembly line. And that process is actually captured by the secretory pathway. And so what we want to do is we want to start off with this idea of creating a protein, first of all, and then kind of following it as it sort of connects these different organelles in line through its process. The first stop that we're going to be running into is the rough endoplasmic reticulum, or the rough ER. Now, the rough ER is so called because it is basically stubbled with ribosomes. So there's lots of ribosomes that are attached to the membranes of the rough ER that kind of gives it this sort of stubbly appearance. And this is the reason why it's called rough. But you could imagine then that if you have ribosomes that are attached to these, then you know that it's doing protein synthesis. But the question is, which proteins? Think about it. There's a lot of proteins out there and they're doing a lot of different jobs, right? Remember in the last chapter, when we talked about the protein structure, we talked about the different functions of the proteins, right? We said some of them are storage proteins, some of them are transport proteins, some of them are enzymes, some of them are defense, right? So there's a lot of different things that proteins and not different types of proteins that you can make. But the thing is, are you making all the proteins here? And the answer is no. You're only making actually 3 major types of proteins with the rough ER. The first one are proteins to be secreted. So anytime you release something outside the cell, that's an act of secretion. So why would you want to secrete a protein? Well, the reason why you'd secrete a protein is to communicate with your neighbor cells, right? So that's how you communicate with each other. You secrete molecules to each other, and your molecules are like your words between cells that coordinate your activities as cells. Very critical function, even on a basic cellular level, for cells as they exist within tissues, right? Tissues have to be coordinated, and the only way you can coordinate is if you're talking to each other. And the only way you do that by secreting molecules to each other. So secreted proteins, proteins that destined to be released outside the cell, are one type of protein that the rough ER will make. The other one is lysosomal proteins. So lysosomal proteins are hydrolytic enzymes, so they're digestive enzymes. So it'll make those. So basically, the lysosome is part of this pathway, this endomembrane system pathway. That's how you actually produce your lysosomes. Yes. Okay. And then the third one are plasma membranes. So these guys are basically membrane proteins that are embedded in the plasma membrane. Notice it didn't say it's making plasma membrane. It says it's making proteins that are sent to the plasma membrane, which basically means they are embedded in the plasma membrane itself. And so those are 3 major types of proteins that you would be getting in the rough ER. Okay, so before we move on to the secondary pathway, we need to finish up the ER piece. And we need to talk a little bit about smooth ER and what the smooth ER does, and we'll kind of get back to the rough ER activities, because that's the process of the secondary pathway. So the smooth ER is called smooth, not because it doesn't have any ribosomes, because it has a lot less, right? So there are some, just not as many. And so as a result, they tend to look smooth, right? Just like smooth membrane. Now you have a couple of different functions with smooth ER. So you have basically lipid synthesis, storage of calcium, and you have detoxification. So let's take a look at lipid synthesis first. So lipid synthesis, basically, when you take a look
at all the lipids in your membranes and all of your organelles in your cell, they're all basically formed of the same structure, phospholipids, right? We studied phospholipids in the last chapter. So you have your phosphate head, but then you have your two fatty acid tails, and you can mix and match those fatty acid tails, right? You can either go saturated, you can go unsaturated. And so you can kind of mix and match the different numbers and amounts of saturated versus unsaturated into your membrane to get different types of consistencies, right? So either solid membrane or more runny-ish type of membrane. And so when you're actually trying to change that up, what you oftentimes will do, you're always kind of maintaining that lipid balance through all of your membranes. Then let's say, for instance, you want to swap out some lipids because you want to maybe add a little bit more solidness to your membrane. So you want to add a little bit more saturated fat. So where do you get those new lipids from then? Well, you get them from the smooth ER, right? So you basically make your new lipids from the smooth ER, and then you traffic those lipids throughout the cell, kind of letting them sort of circulate throughout all the different membranes throughout all the different cells. So all the membrane traffic and the original synthesis of that is coming from the smooth ER. So it's kind of like your lipid synthesis factory. It's the source of new lipids. Now, calcium storage, we're going to get this one from. This is one that you're going to be getting into deeply when you get into AMP1, right? Because this is the basis of muscle contraction. And so in muscle contraction, what you have is in the muscle fiber, which is a muscle cell, you've got a smooth ER, which is actually called sarcoplasmic reticulum, that is basically full of calcium. And that's its job. So its job is to fill up and store calcium. And then when time comes to contract, what you do is you release all the calcium. And the calcium is actually the primary signal for the muscle fibers themselves to actually shorten and then contract. And when you shorten, it's a contraction signal. So it's doing that within the context of being signaled to do so by calcium. So then when you relax your muscle, that is to say you lengthen, you have to mop all that calcium back up, put it all back into the smooth endoplasmic reticulum so that you can release it again the next time you want to contract your muscle, right? So that whole process you'll get into in a lot more depth when you get to the muscle physiology chapter in AMP1. Now, what about detoxification? So detoxification, this is basically something where you have a place, your smooth ER, where you can put like different enzymes that will detoxify toxins that you eat and consume, right? Yeah.
In the sense of like specifics, is it much like turmeric or general? Is there like a specific detox that for your muscles that is the best?
Oh, like some sort of a drink or something like that, or
in this particular situation. Again, all that.
well, for the detoxification, it doesn't have anything to do with your muscles.
Okay.
So it's separate because it's all found in your liver. Right. So the best example for detoxification is actually alcohol consumption, because your body considers alcohol a toxin. And so, like, for instance, if you're not a drinker, right, and you're not usually, you don't drink a lot. How much do you have to drink in order to get a buzz? Yeah, very little, right? And so that's the reason, is because in your liver, basically everything you eat and drink basically gets digested and it gets absorbed into your bloodstream, into your venous bloodstream, and it goes into your liver. Your liver basically processes all, metabolizes everything, defangs everything, detoxifies all the stuff you ate before it releases it to the body, right? So it sends it to the heart for general distribution. So it's kind of like customs, right? It checks everything down to make sure everything is safe for distribution. And so one of the things that liver cells have is a lot of smooth ER, because they've got a lot of enzymes that sort of detoxifying all the stuff that you eat and drink. So if you drink some alcohol, then basically what happens is it doesn't have a lot of enzymes in there to detoxify the alcohol. So you kind of get a little tipsy pretty quickly. But let's imagine that you wanted to start getting buzzed every day. At some point, you start to get acclimated to that, don't you? So your one drink that it took to get you hammered now does nothing for you, right? So what's happened there? Well, what's happened was your liver realizes, oh, I see. So we're taking a drink a day now. So what that means is I'm going to take and I'm going to add more detoxification enzymes into your liver, usually alcohol dehydrogenase. And I'm going to match your level of consumption so that I can detoxify the alcohol that you're drinking. Therefore, you're going to be okay, right? So then what do we do then if we're trying to get our daily buzz? You drink more, right? To which your liver responds by adding more enzymes into the smooth ER. And then you do what? You drink more, right? And then your liver responds, and then you respond, and your liver responds, you respond, right? And eventually, Yeah, right. Right. So eventually you're like, I mean, you got problems, right? Right. This is kind of what we mean when we say the phrase, a person can hold their liquor. What that basically means is their liver has adapted to a chronic consumption of alcohol by loading the smooth ER with these detoxification enzymes. Okay. Yeah. In terms of body mass.
Yeah.
No, she's talking about like you're a lightweight drinker. You can't drink.
Oh yeah. Like me. Like if I just smell alcohol, because I'm like a total teetotaler. I mean, if I smell it, I'll probably just pass out. But right. So I'm okay with that because I honestly can't stand alcohol. But yeah.
I guess my question is.
It's so drinking is a chronic thing. So when you're talking about detoxification, especially with consumption in the liver, you're talking about a chronic exposure, right? So somebody who can drink you under the table, it's pretty obvious that this is something they do on a routine basis.
Or
Even genetics isn't going to do that, right? Because you don't get that. It's an adaptive system. So the fact that you have that many enzymes in your smoothie or in your liver means that your liver has seen this before. So that's that even there's a conditioning in it, right? Now, is there variance in this that's genetic? Yes. I mean, so you can get a genetic variance in this in terms of your ability to do it. But the fact that you can drink somebody under the table is reflective of a chronic behavior. Fetal alcohol syndrome?
Yes, do they already have?
No, because it's an adaptive system. It's all based on what you are feeding your liver. It's all based on what you are eating and consuming, because your liver is seeing everything you eat and consume before it releases it to the heart for distribution. So what your liver is what you are giving it. So your liver is actually what it's responding to is a direct reflection of what you're putting in your mouth. Right. So babies wouldn't have that initially. They would have to ingest it for their liver to be able to respond to that. So the best example of being drunk under the table is one of my favorite scenes in one of my favorite movies. Not the favorite, but one of them is Raiders of the Lost Ark. Right. If you've ever seen Raiders of the Lost Ark, it's been a while now. What is it, like 81 or something like that? It's most of you weren't alive then. But there's a scene in Raiders of the Lost Ark when Indiana goes to, like, I guess he's in Nepal or something like that, goes to find his old flame, Marion. And of course, that didn't go well, right? So she hates him. But the Nazis are on his tail, and they're looking for this medallion that Marion's dad had. And and apparently the scene opens up with Marion in a bar, and she's, got some Sherpa guy, big old guy. Then they're doing a drinking game. So you can see like piles of shot glasses on the table, and they're just downing it, turn it over, right? And it's your turn. And then of course she takes one, downs it, she's good. The big Sherpa guy takes one, and he just keeps on going, right? So he loses. It wasn't so much that. Obviously, she has been doing this for a while, hasn't she? But what's actually more telling about how often she does this is what happens after that. Because after that, Indiana comes in, and then of course, she hates him and loves him at the same time. It's one of those things, right? Because I guess he jilted her or whatever. And they have an argument, and a pretty lucid one. I mean, she takes a couple of shots at Indiana. And I mean, she is completely right there. I mean, she is like right there. And then she's actually coherent enough to actually fight off the Nazis who end up showing up as well. And so, I mean, it's pretty clear. So it's like, wait a minute, if you just drank that much and you are able to function like that, then your liver must be absolutely loaded with enzymes. That means that you do this probably every night. It's drinking games with Marion. Right. So she literally drank the big Sherpa guy under the table.
That's evidence of chronic behavior. Okay. Yeah. So basically, when somebody can hold their liquor, that's in a way kind of a derogatory term. Because it basically what you're saying in sort of a backdoor sort of a way is like, oh, if you can hold your liquor, you've got issues. You have problems. Because that's evidence of chronic behavior. Right.
When we look at like an alcoholic, obviously there's the whole mental process, but like when you get to the point where you become so dependent on it, where it's like if you don't have a drink at any point during the day,
Mhm.
your body starts to
Get the shakes.
shut down.
Yeah, start sweating.
Is that tied into your reserve?
Well, yeah, because what's happening there, once you get up to that level, I mean, what you've done is you've literally like trained your body to anticipate this as a kind of a physiological norm. That's what you've done. And so if you cut cold turkey, which is the reason why alcohol addiction is tricky, and why it can be quite dicey, is because once you've trained your body to sort of essentially sort of view this alternative physiology as the new normal, then when you cut that out, it's like a physiological like trap door opening up underneath you, and it starts to create all these sort of negativities, because now all of a sudden your physiology isn't seeing you going back to normal. It sees you now as being catastrophically abnormal because you told it that dysfunction was function for such a long time that it doesn't really know what it's supposed to do anymore. And that's where you get all those negative adverse withdrawal effects. And that's true for most addictions like that. Is once you kind of get to that chronic level of addiction, it trains your physiology to say this is the new normal. So when you go off of that, then it's like a catastrophic shift back down to normal. What we know is normal, but your body is thinking, oh my gosh, this is like, it's all falling apart now. And so it's like, it doesn't know what to do. And it can be quite ugly and quite dangerous, which is the reason why for a lot of recreational drugs, you actually have to have a very medically directed step down procedure to take yourself off, or you could actually harm yourself. Lorraine first and then Jared. Caffeine. Yeah, caffeine, I mean, it's addictive, right? To a lesser extent. Anybody that had a caffeine headache, that's basically it. That's kind of a mild withdrawal symptom. But I mean, it's pretty much done within a day, right? So yeah, and caffeine's not as ugly as some other things. Nicotine can be quite bad as well. It's much worse than people give it credit for, actually. Okay, Jared.
That's why you see a lot of heroin users who die because they'll get clean and then they'll go back and do the same amount they thought they used to be able to do.
Yep.
But now it's the color.
Yeah, they've recalibrated themselves, so they can't go back to their left off dose. They would have to act like they're just starting over again and ramp themselves back up. Yep. So that's a huge problem.
For like a trade of booze, like a huge consumption of sugar, would that be like?
Oh, the sugar itself,
Yeah, like if you're like.
because it gives you like a little energy buzz.
Yeah.
Not so much. I mean, sugar is addictive because it's one of our mammalian cravings, but it's not the same sort of a thing.
Would you have, like, if you consume a bunch of sugar, would you have like a high level of enzyme?
Well, sugar's not a toxin, right? So your body processes it differently. Drugs typically are toxins. And so typically the smoothie R is only focused on toxins. So you process sugar differently.
Why, like, I guess this is just, I'm sorry to continue to derail further, but I guess my question is, why can some like homeless people down here just get pot, like then drugs, like it's not, and be completely fine, and then like if like you just even get a whiff or like a smell of it, if you have
Yeah,
That's not true. Those videos, those cops.
yeah. I mean, the problem with different drugs is, and it's because this is like a general pattern that we see, but different drugs have a lot of nuances to them.
So like not every drug behaves the same. Your body doesn't behave the same to every drug. So it has a very different response to them. And all of that kind of factors in it because every single drug has a very specific physiology to it. And so even though there's like general patterns that we see in kind of drug addiction, things like that, for more of a specific treatment, like a medical treatment of it, you would have to actually be like, okay, we need to really understand what is the physiological pathway that this particular drug is affecting, because there's nuances and specificity in the physiology that would actually crop up. Right. Yeah. Yeah, because some have more of a depressant activity to it, right? Uppers and downers, right? So, you know, and then you kind of get cross-threading where you have two contradictory things going on in your body. And so basically it's like taffy pulling your physiology, and then you just kind of, yeah. Yeah. So, okay, so we can actually tell what a cell does for a living by just looking at the ratio of ER, so rough to smooth, right? So for instance, if you have a gland, right, what do glands do for a living? They secrete stuff, right? So what would the ratio be for rough to smooth? Would it be high or low? So its job is to, so you would expect this ratio to be high or low? High, right? Because you have a ton of rough, right? Because your job is to secrete. You're making a lot of the secretory proteins. What about the liver? We just talked about that one. So this one would be lower, right? Because you'd have more SER instead of RER, right? Because you're detoxifying. Well, it's because that's what it does for a living, right? Notice the liver, one of its big jobs is detoxification. So that means you're smooth dominated. So as a liver cell, you're not really going to be secreting stuff, right? You're not going to be making a ton of lysosomes. You'll have membrane protein, so your rough will be there. It'll be doing stuff. But really what you're doing is smooth ER. You're detoxifying. That's what you do for a living. That's your thing, right? Okay. So here's kind of what it looks like structurally. So you can see the rough ER basically has this kind of labyrinthine look to it. You can see these like little ribosomes that are sort of tethered to the side. And then you kind of see this like little pucker outward where it kind of folds into the smooth ER, which kind of has a slightly different structure. So you can kind of see when you kind of cut it in cross section, it looks like little tubules. So you're kind of looking down the cross section of the little tubules themselves. And when you take a look at the transmission electron micrograph here, you can see the different pieces, right? So this would be your rough over here. So you can see the individual like ribosomes here, the little black dots. And then here you can kind of see this is your smooth ER. So you can see there's not a lot of ribosomes over there on that side of things. Now, the other thing that I want to do is I want to basically take a look at its relationship, the rough ER, the endothelial reticulum's relationship to the nucleus. Because remember we said the nucleus has a double membrane system, right? So you have an inner membrane and an outer membrane. So actually, when you take a look at the rough ER, it's actually continuous with the outer membrane of the nucleus. Because what happens is the outer membrane basically just sort of evaginates. And then when it does, it kind of folds down and kind of forms this little flap, which is the first labyrinth of the rough ER. Then this will do the same thing. Yes, right. So you're kind of just basically extending it off of the nucleus, right? So a couple of things to note here is the lumen of the rough ER is continuous with the space of the nuclear envelope. And not only that, but you have the same thing to make the smooth, right? So the idea is if you have a nucleus, then you have basically a mechanism to basically make the rough and the smooth. Right. So this is kind of nice from a kind of an origins point of view, right? Whereas like, where does all this stuff come from? Well, you're not looking at three different events. You're looking at one event that just kind of morphs into the rough which then morphs into the smooth, right? So that's basically what we've got here. And so ultimately what will happen is you'll have a ribosome that's tethered to the rough ER. It'll make the protein, injecting it into the lumen, and then it'll fold up inside the lumen. And then when it's ready to move on to the next stage, what's going to happen is it'll basically swell and it'll pinch off into this like little bubble, membrane bubble. This would be your transport vesicle. Remember, this is your membrane bubble. That houses the crude protein. Its next stop, the Golgi. So the transport vesicle is going to be taking this crude protein to the Golgi, which is a flattened system of membranes. So just kind of all flattened, like a little stack of pancakes. And the Golgi has those three main processes, right? Modify, that's basically the molecule synthesis. And then it's got to pack and distribute, right? So that is the sorting and the shipping. Then it's got to send it out to its location where its destination goes. It has two different faces on it. The cis face, which is the same side, trans face, which is different or across. And the reference point here is the nucleus. So it's on the same side of the nucleus. So this is the nucleus side. And the trans side is the membrane side. And so ultimately your vesicle will transport it to its destination. So you can see here's a transport vesicle that will then fuse, like this guy right here, with the cis face. That's the nucleus. So the nucleus is over here. And then it'll dump its crude protein into the Golgi, where the Golgi will modify it, sort it all out according to the type of protein that it is. And then once it's done with it and it's finished, it'll basically put it into another vesicle on the trans face. And then it'll pinch this off and it'll become a secretory vesicle, right? Now this is basically the membrane bubble that houses the final finished protein. This is protein that's ready to be released. And this is kind of what it looks like. So you can see here's your cis face and here's your trans face. It's kind of like a receiving dock on the cis face and a shipping dock on the trans face. And so this will be making modifications to those proteins in basically what is the secretory pathway. So basically what I want you to do is I want you to take a look at this. This is essentially what we just talked about, only it's kind of in one big long flowing picture. So here you can see proteins that are being made. These are the secreted proteins here that are being made and put into a vesicle. You also have membrane proteins here that are actually bound to the membrane itself. This transport vesicle will break off, so holding its crude protein and the membrane embedded proteins in the membrane itself. It'll dock on the cis face and it'll dump all that crude protein into the lumen of the Golgi, where it'll be modified and perfected and finalized. And then the membrane protein will basically be embedded in the membranes and it'll also be modified as well. And then at the end, you'll notice you'll pinch off your final protein into your secretory vesicle. And then you're going to go to the cell membrane where you'll fuse with the cell membrane, like two soap bubbles fusing together. The vesicle will fuse with the cell membrane and it'll dump all the contents to the outside of the cell. That's secretion. So this whole thing right here is the secretory pathway, which ties together a good huge chunk of the endomembrane system. So the other thing that we want to take a look at is when we take a look at a secretory vesicle, we have these secreted proteins on the inside, but the membrane proteins will still be embedded in the vesicle. So as this vesicle travels to and fuses with the plasma membrane, it turns kind of inside out, and all these little secreted proteins will basically exit. The membrane proteins will still be in the membrane. But what will happen is the vesicle turns inside out. So then eventually those membrane embedded proteins will be on the outside of the cell. And that's how you move proteins to the outside of the cell, to the periphery of the cell. You load them up in a vesicle first.
When you fuse them with the membrane, it incorporates with the plasma membrane. Yes.
Like shoot out of.
No, it's slow. It doesn't like, yeah, it's not like a shotgun. It's slow. It depends on the need. Yeah, it depends on the need and how much protein you're making and what the situation is. It's very adaptive. So lysosomes, right? The reason why we're talking about lysosomes is because basically lysosomes look exactly the same as secretory vesicles. They're membrane bubbles. The only thing that differs is what's in them. So what's in them? Enzymes, hydrolytic enzymes that do hydrolysis, right? So basically what they're doing is they're breaking down macromolecules. They're breaking stuff down. Remember, that's one of the proteins that the rough ER is making. So in this particular case, the Golgi's job is to identify those hydrolytic enzymes and to put them separately into their own separate little membrane bubble. That becomes the lysosome. You hang on to that one, right? This is like the garbage disposal of the cell. You don't want to release digestive enzymes to the outside world. Why? Because you'll digest yourself, right? You'll digest your neighboring cells. That's not a good thing, right? So ultimately then what this does is it creates kind of almost like a cellular stomach, right? This is a reservoir where you have all your digestive enzymes and allows you to be able to digest foreign things. So you can bring in foreign stuff and engulf them, and you can eat them by a process called phagocytosis, which is essentially, phago means to eat, cyte means the cell, and osis is the condition of. So this is the condition of cell eating. So phagocytosis is basically a situation where a cell will kind of wrap itself around some sort of food particle, in this case, like your white blood cells, engulfing a bacterium, will basically ooze around this bacterium and then basically fuse this with lysosomes and then digest everything. So all those hydrolytic enzymes will just tear this bacteria apart, and then you'll be able to use like amino acids and things like that for your own proteins. So here's the two ways that a lysosome is used, right? So the first one is recycling, and the second one is cell eating. So for instance, if you have like an old organelle, like in this case, an old mitochondria, and you need to break it all down, you want to reuse its parts. You want those amino acids back so you can make your own proteins. Then all you have to do is just fuse a lysosome with it. And then what will happen is those hydrolytic enzymes will just chop it all up, tear all those proteins and amino acids, those nucleic acids into nucleotides, and you can reuse those parts, materials to build your own proteins with. That's if you're like multicellular, right? Like us, for instance, we do the recycling thing a lot. If you're a single celled organism, you don't have a stomach, then you have to figure out a way to eat. The way you eat is through phagocytosis. So basically what's going to happen is you're going to suck in little food particles, surrounding them with your membrane. And then once you pinch that off, it's going to create a little food vesicle. And then you just fuse that with lysosomes. Your hydrolytic enzymes will chop them all up, and then you can use those amino acids for your own benefit. Okay. So if you're a single celled organism, this is the way you eat, because you don't have a stomach. There's other microbodies, which are just basically other little membrane bubbles, which all differ based on what's in them. And one of those microbodies is what's called a peroxisome. So a peroxisome basically has enzymes in it that basically detoxifies the oxidative byproducts of oxygen consumption. So oxygen consumption is actually quite lethal. It can be as lethal as cyanide, actually. The only reason why we can survive is because we've got enzymes in our peroxisomes that detoxifies these really nasty oxidative byproducts. We're talking about things like free radicals and superoxides. So basically what these do is these have unpaired electrons, which makes them oxidative. So they'll take an electron from your tissue, which will damage your tissue, or your DNA, which will mutate your DNA. It's one of the reasons why you want to eat a lot of antioxidants, like in blueberries, for instance, is because those are molecules that will basically offer their electrons to these free radical superoxides instead of it taking it from your tissues, right? Thereby reducing the likelihood of you developing cancer. right? It doesn't necessarily won't develop cancer. It did because cancer is all random, right? It just, you can still be unlucky. You can eat all the blueberries you want, but you can still be unlucky, right? But it just reduces the likelihood because you have all these antioxidants that are sort of throwing themselves on the sword, so to speak, right? Taking the hit instead of your tissues. Hydrogen peroxide is a good example. That's an oxidative one, but we have in our peroxisomes an enzyme called catalase, which will take peroxide, turn it into water, harmless, and oxygen, also harmless. So a detoxified peroxide. Yeah. No, not necessarily, because they've all basically grown up in the same oxygenated area. So our tissues are pretty similar that way. So the other one that we have is an organelle. It's not a membrane-bound organelle. It's actually a protein machine. It's called the proteasome. So this basically, what this does for a living is it digests protein. And this is to basically maintain and regulate your protein, your proteasome, which is all the proteins in your body, right? Because you don't need all your proteins all the time. You need protein X, a lot of it at this particular moment. But then when you're done with that moment, you need to bring that back down to normal levels. So this allows you to be able to regulate your protein levels, to increase them and then to decrease them in a very functional way. It also allows you to be able to get rid of misfolded and damaged proteins, right? So basically what happens is if you have misfolded and damaged proteins, then the first thing that happens is you tag this protein for destruction by adding what's called a ubiquitin to it. It's a molecule that's added to the protein. It's like the mark of death, right? You've been marked for degradation at this particular point. And there's an enzyme that does that, ubiquitinase, that does that. And then once you've been ubiquitinated, then what happens is you're going to thread this protein through the proteasome, and it's like a big meat grinder. It's just basically going to grind up that protein, and then on the outside, you're going to get your amino acids back. And then you can go make more protein. No, it's recycling it. Yep, exactly. And so this is a really important kind of an idea. And then the last thing I want to talk about before we move on to the next section is going to be the vacuoles, right? So there's three major types of vacuoles. There's obviously the big central vacuole that basically is involved in water balance. So this is basically where plants will accumulate water when you're adding lots of water to them, or when you're taking water out of the cell. Basically, it's associated with that. The contractile vacuole is also about water balance. And this is very simple. It's like your bladder, right? The contractile vacuole, like for instance, in a paramecium, basically will alternate be in between an empty state and a full state. So what happens is, just like your bladder, the contractile vacuole has these like little tentacles, these channels that radiate throughout the cell. And they basically siphon off water. So water that's coming into the cell, that's flooding into the cell, that water goes through these little channels and fills up this little bladder-like vacuole. When it fills up, basically it contracts just like your bladder. And then what it does is it squirts that water outside the cell. So the way that the paramecium actually balances its water is by bailing water. Literally, as the water floods into it, it basically uses the contractile vacuoles to squirt the water out of it, and it soaks it back up like a sponge, and it squirts it back out, and it soaks it back up like a sponge, and it squirts it back out. That's how it maintains its water. And then, of course, you have storage vacuoles like the food vacuole that we saw before, where you're storing food in it. Okay. Okay. So that's the end of the second group. We'll begin next time with the last two, starting with the energy group.